Hello everyone. Let's get started. Today we're going to be talking about pathogens and how they spread. If you recall from earlier in the course, we talked a little bit about the history of the beliefs around infectious disease. We talked about how the earliest times in man's recorded history, people blamed disease on the gods, on spirits, being displeased with them and their actions, and how people had to pray and sacrifice to the gods in order to prevent disease outbreaks from occurring. We said that during the time of Hippocrates, people blamed imbalances within what were called the four humors of the body for the occurrence of disease. And if you recall, these humors were the blood and the phlegm, the black bile and the yellow bile. Later on, in the early 1800s, arose Miasma Theory and another called contingent contagionism. Miasma Theory, if you recall, is the theory that bad smells and filth and decay, or what was referred to as bad air, was the cause of disease. So it was pollution from swamps and rotting materials and poor hygiene and so on that created these horrible smells and this bad air. And people believed that it was exposure to the bad air that caused disease. Miasma theory actually did not state that disease could be passed person to person, but rather anybody who was exposed to the bad air would get the disease. Contingent contagionism was a little bit different. The bad air was still the cause of the disease, but people were starting to believe that once a person had that disease, it could be passed from person to person as long as both people were in that bad, unhealthy environment. Another theory of the same time was Zymotic Theory, and it was the Zymotic Theory that we associate with the idea of spontaneous generation. Remember, this spontaneous generation theory stated that living things could arise from non-living things, again, out of dirty and filthy environments. So, for example, fleas could spontaneously generate out of dirt and dust, and maggots could spontaneously generate out of rotting flesh. The idea was that decomposing things could somehow produce forces that would lead to the generation of other living organisms that would then go on to cause disease. It wasn't until the middle part of the 1800s that Louis Pasteur and other scientists developed what we now think of as the germ theory. And the germ theory stated that disease was caused by microorganisms, living things that were too small to be seen with the naked eye, and things that could get into the body and grow and reproduce and cause disease. Recall that Pasteur was actually a chemist. He did a lot of very important things and made a lot of contributions to science. He outlined the process of fermentation, for example. He contributed again to the development of germ theory. He also developed the earliest vaccines, including vaccines for anthrax and rabies. It was also Pasteur who developed the process of pasteurization. And remember, that process was originally developed to help preserve French wines. A couple of other names that we should know from history when it comes to infectious diseases and how pathogens are spread include Joseph Lister, who is considered to be the father of antiseptic medicine. Lister was interested in why so many women who gave birth would come down with infections and die afterwards, and why patients who underwent surgery would often come down with infections and die. It was Lister who recognized what was referred to as ward fever, this this phenomenon where people who had come out of surgery, people who were in the post-surgical ward of a hospital, would often die because they had developed infections. And of course, we know that those infections came from a lack of aseptic technique during surgery. So it was Lister who figured out that infection could come from the environment. And he was a proponent of using hand washing to help prevent these kinds of infections. Lister actually used a chemical called carbolic acid, which is not something you would want to use every day to wash your hands, but it does the trick. It disinfects the hands, and Lister also used it on surgical instruments and other materials that were used on these hospital wards to help reduce the number of deaths that were associated with infection. So Lister pioneered the use of clean, sterile technique during hospital procedures and the handling of patients. Ignazio Vice is the physician who is credited with pioneering the process of washing your hands as a routine part of medical practice. It's Semmelweis who determined that the physician was often the one who was passing the infectious disease from patient to patient to patient, because the physician would pick up this disease on their hands when they were working with one patient and then walk over and transfer that disease to the next patient with those dirty hands. So hand washing became part of just the routine of a physician, thanks to Semmelweis's writings. It's interesting because hand washing has been in the news lately with the epidemic of COVID-19. Hand washing is still the number one way to reduce the transmission of infectious disease, particularly infectious respiratory diseases like colds and flu and this novel coronavirus called SARS-CoV-2. And that brings us to Robert Koch. Koch was a scientist who was working during the time that all of these competing theories about disease transmission were in society. And it was Koch who developed a system for determining exactly what causative agent of any particular infectious disease was causing the disease. Koch came up with a series of what are known as postulates, a step-by-step guide for a scientist to determine whether or not a particular microbe caused a particular infectious disease. In fact, it was Koch who first showed that Bacillus anthracis was the bacterium that caused the disease anthrax. And he did that by following these postulates. So let's take a look at Koch's postulates, the step-by-step guide for figuring out whether or not a microbe causes a particular disease. The first thing that has to be done is that pathogen has to be isolated from an animal or a human that has this particular infectious disease. The pathogen then has to be brought into the laboratory and grown in the laboratory as a culture. So of course, in Koch's day, this meant that bacteria were the microbes that were being attributed with diseases. Koch would not have had the ability to culture viruses, in other words, in the laboratory. The next step was to inoculate that pure culture into a healthy animal specimen, an animal of the same species, and create that same disease, pass along that same disease to this new animal. And then, importantly, you had to re-isolate that same pathogen out of that newly infected animal, bring it back into the laboratory, and culture it again, and make sure that you could match the pathogen out of the second animal with the pathogen out of the first animal. On this slide, we can see a diagram of this process that Koch was using that led to the development of Koch's postulates. Up at the top here, you can see a drawing of a rodent, a rat maybe, that is infected with some disease. And the pathogenic or the suspected pathogen would be isolated out of this diseased animal and brought into the laboratory where it would be cultured. Once it was grown up in the laboratory, it would be used to infect a healthy animal that would then come down with the disease. The pathogen would then have to be re-isolated out of this animal and grown up in the laboratory again. And then a determination had to be made that this microbe from this second animal matched the microbe that was isolated from the first animal. And if it did, then you had satisfied the postulates, and you could say that this is the microbe that causes this particular disease.